Welcome everyone to our second lecture of AEM341 or General Microbiology. Today I'm going to be telling you guys a bit more about the history of microbiology, how we came to know what we know today, and how the science as a whole developed through the last couple of hundred years. So for most of human history, people had no idea how disease was spread, and they believed some really crazy things. So for example, here, this sketch, sorry, let me just get my pointer. There we go. Here in this sketch, you can see this is actually a representation of the cholera epidemic of the 19th century that depicts the spread of disease in the form of miasma, also known as poisonous, foul-smelling, bad air. So people essentially believe that bad smells was how the disease was spread. So vapors and bad air. They had no idea of the existence of microbes or pathogens or any of that. So the first, let's say, foray into microbiology was actually done by Robert Koch, and he invented, so that's Robert Koch over there, and he invented this microscope and illumination system with which he viewed many things, but most notably he viewed cells of cork, which you can see that was his depiction of what he saw. That's what he drew. And he said of cork that I could exceedingly plainly perceive it to be all perforated and porous, much like a honeycomb, but that the pores of it were not regular. These pores of cells were indeed the first microbial pores I ever saw, and perhaps that were ever seen, for I had not met with any writer or person that had made any mention of them before this. So essentially, what Koch had discovered were plant cells. So looking at cork today, that's essentially what you would see. And he saw these cell walls, and he in fact actually coined the term cells, the box-like structures in the cork that he saw, because they reminded him of cells in a monastery. He also reported seeing similar structures in wood and other plants. Cork cells are, of course, much larger than bacteria, so his microscopes could not view most microorganisms. However, his work was an important contribution to microbiology because it started the idea, the notion, that there are these biological structures that are simply too small for the naked eye and for humans then to see without the aid of a microscope system. Following the work by Koch, Antonie van Leeuwenhoek was the next big name in microbiology, also known as he later became known as the father of microbiology. So he was a merchant and only an amateur scientist, and he got interested in making magnifying glasses to see if he could see the quality of thread more clearly. And he reported seeing, eventually that changed into him describing wee animicules in rainwater infusions and tooth scrapings, because he had hundreds of these little microscopes. You can see that's what it would look like. And this is how it would function with your sample translator at the bottom, how you would move the thing, the focus knob, the sample holder being this pin over here, and then the lens. You can just imagine the strain moving your sample through a little lens or hole that small would place on your eye. It would have been pretty bad. So at the end of his life, he ended up owning like approximately 400 of these little microscopes. And notably, he was actually able to get the magnification up to 300 times, which is really quite good considering the simple structure of these microscopes. He described seeing the movement or seeing movement. These was of course the movement of the microbes as they moved through whatever he was looking at. And then he observed microbes in rainwater, feces, dental plaque, and then he drew the organisms that he saw. And that's what this is. So these are the rudimentary drawings that he made of the things that he saw through his microscopes. Notably, however, he didn't write any books. His discoveries came to light through correspondence with the Royal Society, which published his letters. So at this point, humanity knew that there were living organisms we couldn't see, But scientists and everyone else continue to believe that organisms just spontaneously arose from nothing. This is also known as the spontaneous generation body of thought. So the Greek philosopher Aristotle was one of the earliest recorded scholars to articulate the theory of to articulate the notion of thought of spontaneous generation. I'll explain why it's not a theory in a second. The notion that life can arise from non-living matter. Aristotle proposed that life arose from non-living material if the material contained what he called pneuma, which means vital heat. As evidence, he noted several instances of the appearance of animals from environments previously devoid of such animals, such as the seemingly sudden appearance of fish in a new puddle of water, which to him supported the notion that life arose from non-living things. They also noted that, or thought at that point, that, for example, moist soil gave rise to toads and snakes, that flies were born of manure, and that maggots are born from corpses. Because they couldn't see the actual processes, didn't understand them in depth. They saw, well, here was a piece of meat, and now a couple of days later, well, now it has maggots on it, so the maggots must come from the meat, or it must arrive from this non-living corpse. And the spontaneous generation body of thought was widely accepted until Francesco Redi, who was a physician, set out to challenge this idea. Note the wording here that I specifically call it a body of thought and not a theory. This is essentially to segue into how what constitutes a scientific theory. People who don't know any better often like to say that, for example, with a theory of evolution, oh, it's just a theory, it's not a fact, it's just a statement someone made. And that is not the case, not at all. For something to constitute an accepted scientific theory, as the case with the theory of evolution, it has to meet certain criteria, which means it cannot just simply be a statement. I can't just say anything and say, well, it's my theory. Scientifically speaking, that is not correct. So to go a bit more into depth of that, the University of California, Berkeley defines a theory as a broad natural explanation for a wide range of phenomena. Theories are concise, coherent, systematic, predictive, and broadly applicable, often integrating and generalizing many hypotheses. So you can find many definitions of what a theory actually is, or what actually constitutes a scientific theory, but there are some commonalities. Most of these definitions tend to agree that a theory needs to be substantiated, so you need to find actual evidence supporting your claim. It needs to be explanatory. You have to be able to explain an actual phenomenon with it. They have to be predictive, so you have to be able to apply your theory and predict what is likely to happen given a certain scenario, and they have to be testable. You cannot claim something that cannot be tested. So for something to be a scientific theory, it has to adhere to those principles or to those factors. So the entire take-home message from that little tangent is that a scientific theory is not an untested claim made by someone. It is very much a process to have something established as an accepted scientific theory. So going back to spontaneous generation, Francesco Redi initially conducted experiments using two jars where he would place meat in both, and then he would close one close one off and leave the other one open. And then after a few days, he saw, oh, wow, there are only maggots on the open jar. But then people said, no, that doesn't disprove spontaneous generation, because spontaneous generation requires fresh air. So he said, well, okay. And then he set it up with, he repeated the experiment using a jar with mesh covering, so air could still get in there. So he challenged the idea that the maggots on the meat is spontaneously generating from the meat. And thus, in this instance, what he found that the maggots were only on the mesh, not on the meat. And so This was the first major experiment used to challenge spontaneous generation. And then he used these experiments to support the pre-existence theory, which was put forth by the church at the time, which maintained that living things originated from parents. So that was his contribution to microbiology. And then, okay, so people then went to the notion that, okay, so maybe larger animals can't spontaneously generate, but animalcules that are so simple and so small, surely they just generate from non-living materials. So here you can see the maggot. So yeah, okay, it's a big, complex creature. It's big compared to the microbes, at least. So yeah, okay, so they essentially said, yeah, this couldn't just spontaneously generate, but surely these little small organisms, or animalcules as they were known, can just pop into existence. And so then spontaneous generation was accepted for another 200 years, specifically regarding microorganisms. And then in 1858, Rudolf Virchow, I'm not exactly sure how to pronounce that name, stated that life can only come from pre-existing life, but he couldn't test it. And then a few years later, Louis Pasteur, who was a French scientist, set out to test spontaneous generation. Okay, so Pasteur then designed a simple procedure to test if sterile nutrient broth could spontaneously generate microbial life. So to set this up, he essentially made two experiments. These experiments are the one at the top and the one at the bottom. So first, he boiled this broth, right? On the broth of both flasks. So this was the flask with the bent neck. This S-shaped bent neck is vitally important. And then the flask with the normal neck. Both of these flasks were then subject to boiling. So he sterilized the broth at that point. So he did that to kill the existing microbes, and then he left both of the broths to sit at room temperature and expose them to the air. So he opened these corks, he left them like that. And then after several weeks, he noticed that the broth in the straight neck flask, so this broth, the one at the top, was discolored and cloudy, while the broth in the curved neck flask had not changed. So you can take a couple of seconds to think, what do you think he concluded from this? So go ahead and pause the video and think about it for yourself for a second if you want. But okay. So what he actually concluded was that the germs in the air, the microbes in the air, were able to fall unobstructed down into this flask, but not so much into this flask because they would get trapped in the S-shaped neck. So that's why this one became cloudy and this one did not. So essentially, this is a better representation. So here comes a little germ and it just plops down into the broth, and then it becomes cloudy and it changes because of the contamination by the microbes that were in the air. And in the second instance, the microbe will fall into the open flask, but it'll be trapped in the neck of the flask and thus not contaminate the broth. So if spontaneous generation had been a real phenomenon, he argued that the broth in both of the flasks would have been cloudy and would have been contaminated because the microbe would have then just spontaneously generated in there. But the curved neck flask never became infected, indicating that germs could only come from other germs. His experiment also held two other very important implications. The first was that microbes can be in the air, and the second was that microbes can be destroyed by heat. He was successfully able to kill them off at the start by boiling the broth. These were discoveries essentially constituted the beginning of aseptic techniques, procedures that prevent contamination by unwanted microorganisms. So Pasteur's experiment also led to the development of preservation and sterilization techniques. For example, all of you probably buy pasteurized milk, and that Pasteur that you see in pasteurization comes from his name. It comes from Louis Pasteur. So pasteurization is a process in which packaged and non-packaged foods, specifically milks and fruit juice, are treated with mild heat, usually less than 100 degrees Celsius, to eliminate pathogens and extend shelf life. So here you can see the fresh milk is placed in, it is then heated to sub 100 degrees, and then cooled, and then bottled and shipped out to stores. And this simple process extends the life because it neutralizes or kills off many of the microbes that are found in the milk originally. So that essentially led to Pasteur then saying, officially, I recommend pasteurization of milk, but I still like drinking it straight from the udder. Okay, so now, at this point, before the 1880s, we've known about animalcules for about 200 years, and everyone still thinks that disease is caused by vapors and bad air. No one has linked microbes to the causing of disease yet. And they've essentially said that these vapors that cause these sickness, this miasma, can be identified by bad smells, and then they are thought to arise from dead and decaying organic matter. So the miasma theory is an obsolete medical theory that held that diseases such as cholera, chlamydia, or the Black Death were caused by miasma, a noxious form of bad air, and that was also known as night air. So something else that also contributed to this, I mean, here's another depiction of the bad air. So you can see the air forming from the chimneys that forms this bad miasma, and people believed that was what was making people sick with cholera or the Black Death. People also often believed that disease was a punishment for your crimes or your misdeeds, kind of like holy intervention. And when a whole village would fall ill, people would often blame demons as opposed to just, investigating the actual causes of why these people got sick. So let's move on to some plague doctors. So because people believed that smelly vapors could catch in the fibers of their clothing and transmit the disease, one of the first plague doctors, or the person who designed their outfit at least, I'm not sure how to pronounce his name, Delorme, designed a uniform of waxed, that comprised a waxed leather coat, leggings, boots, and gloves intended to deflect miasma, so bad air, from head to toe. The suit was then coated in sweet, which is a hard white animal fat, to repel bodily fluids. And the plague doctor also donned the prominent black hat over there to indicate that they were, in fact, still a doctor. These doctors carried a long wooden stick, you can see it over there and over there, which he used to communicate with patients. Essentially, the mask would make it difficult for them to speak, so they would use this stick to essentially prod and poke, a person, and to tell him to do this or to do that. And then they also used the stick to examine these people and to ward off the more desperate and aggressive ones. So essentially, you could come running up to the doctor and then he would beat you with his stick. By other accounts, the patients believed that the plague to be a punishment from God, as I said, like holy retribution or whatever you want to call it, and requested that the plague doctor whip them in repentance. So you could go walking down the street and see your local doctor just beating the snot out of a sick person with his little cane. So the foul-smelling air was also combated with sweet herbs and spices like camphor, mint, cloves, and myrrh, stuffed into the mask with a curved bird-like beak. So that was the reason for the beak. They would stuff this full of strong smelling herbs to kind of combat the miasma. Remember, they thought that it was the foul air, the stench that was essentially making people sick. So if they couldn't smell the stench, they couldn't get sick, was their thinking at the time. And sometimes the herbs also set aflame before they were put in the mask, so the smoke could further protect the plague doctor. You can see that in this artist representation over here. Yeah, I'm not gonna lie. If my doctor, shows up with a flaming beak bird mask and full black robes and a fighting stick or a beating stick, pretty sure I'd never fake an illness in my life ever. Be like, yeah, now I'll go to school, mom, every day. So eventually, though, there was some progress on this front. So Ignaz Semmelweis during the 1840s started the notion of the germ theory of disease, or at least got us on the right track, got scientists on the right track. So he worked in the Vienna hospital where 30% during certain months, specifically the Decembers and stuff, 30% of the women delivering children were dying from childbirth fever. That's that puerperal fever. So 30%, almost a third of the women were dying because of that fever. And we now know that this fever is caused by an infection, most often of the placental site within the uterus, and this can lead to sepsis if the infection reaches the bloodstream. So, so how he arrived at his proposal of hand washing with chlorinated lime, it's quite an interesting story. So his breakthrough occurred in 1847. Following the death of one of his friends, Jacob Kochka, who was poked by a student's scalpel while performing a post-mortem examination. So his friend was stabbed with a knife that was contaminated by a post-mortem autopsy, essentially. And then this Kochka's own autopsy showed a pathology similar to that of the women who were dying from puerperal fever. So Semmelweis immediately proposed the connection between cadaveric contamination and puerperal fever. He forced everyone to wash their hands before and after patient contact, and this decreased mortality from childbirth fever to around 1% during certain time periods. However, Semmelweis's observations conflicted with the established scientific and medical opinions of the time, and his ideas were largely rejected by the medical community. He could offer no acceptable scientific explanation for his findings, and some doctors were even offended at the suggestion that they should wash their hands and mocked him for it. In 1865, the increasingly outspoken Semmelweis supposedly suffered a nervous breakdown and was committed to an asylum by his colleagues. He died 14 days later after being beaten by the guards from a gangrenous wound on his right hand, which might have been caused by the beating. So it's a very sad end to someone who had such a impactful contribution, or who were actually steering the world down the correct path. So then we move on to the 1860s with Joseph Lister. So Lister was aware of the work of both Pasteur and Semmelweis, and he decided that if clean hands are good, clean surgical instruments are also probably good. So he essentially started to sterilize his surgical equipment before cutting people open with it. So he disinfected his instruments with carbolic acid, now known as phenol. And these people made some interesting leaps, but he essentially assumed that this would be an adequate disinfectant because it was used to treat the stench from fields irrigated with sewage waste. And he also surmised that it was probably safe to use because there were no ill effects in the livestock that were grazing on these treated fields. But luckily enough, he arrived, his practice of sanitizing his equipment before use was greatly reduced infection and death following surgery, and other surgeons quickly adopted similar practices. frustratingly enough, though, at this point, that there was still not enough evidence to establish the germ theory of disease. Lister and Semmelweis provided good evidence, good anecdotal and circumstantial evidence for germ theory, but did not provide definitive proof. These experiments were very much circumstantial, meaning they were not properly controlled. They weren't conducted in a controlled environment where other variables couldn't potentially interfere with the results. So thus, we needed someone to come along with a properly controlled investigation. Now we get to Robert Koch, or Koch. Robert Koch was a German physician who began researching microbiology. And he did a lot of work with numerous microorganisms and diseases. And he also established the germ theory of disease. So the germ theory of disease is the currently accepted scientific theory for many diseases. And it states that microorganisms known as pathogens or germs can lead to disease. These small organisms that are too small to see without magnification invade humans or other animals and other living hosts. Their growth and reproduction within their host can then cause disease. Germ may refer not only just to a bacterium, but to any type of microorganism, such as a protist or a fungi, and even non-living pathogens that can cause disease, such as viruses, prions, and viroids. So Robert Koch was widely known for his work with anthrax, where he discovered the causative agent of the fatal disease to be Bacillus anthracis. He also discovered the formation of spores in anthrax bacteria, which could remain dormant under specific conditions. These spores have historically been used in certain assassination attempts, notably. However, under optimal conditions, the spores were activated and caused disease. To determine this causative agent, he derived fixed bacterial cultures onto glass slides. He used dyes to stain the cultures and observed them through a microscope. His work with anthrax is notable in that he was the first to link a specific microorganism with a specific disease. Really important. So he was able to trace that the Bacillus anthracis was the causative organism for anthrax. His work then thus led to the rejection of spontaneous generation and supporting and the formulation of the theory of germ theory of disease. So he also was responsible for formulating Koch's postulates, really important, we'll get to that in a second. But essentially, what Koch did is he saw that sick animals had bacteria in their blood. That was not enough to prove that the bacteria was causing the disease, right? So he saw a healthy organism, took a sample, the red blood cells, and then he found that the causative agent was absent. Then he took a deceased organism and found that there was a suspected agent, and then the causative agent was present. But this was not enough to prove that this bacteria was actually causing the disease. I mean, it could have just been the result of the disease. So the disease could be causing the microbe just at this point, just as easily as the microbe could have been causing the disease. So that's why he then attempted to isolate this disease, or the microbe that causes the disease, and then reintroduce it into an animal to see if the animal will become ill. And this process that he went through led to the development of Koch's postulates. So Koch's postulates are still used today to identify the causative agent of a disease. So the first of these postulates states that the microorganism must be found in all cases of the disease. So obviously, if the microbe is what's causing the illness, if someone has that illness, they must have the microbe. The microbe must be isolated from the host and grown in pure culture. It must then reproduce the original disease when it is injected into a susceptible host. So once you've isolated your organism that you're investigating, that you are suspecting of causing this disease, if you introduce that microbe into a new animal that is free of disease, it has to cause the disease. And then, following this reinfection, it must also be found in the experimental host that was infected. So if you introduce the microbe and the organism becomes sick, but then you test that sick organism and that microbe is not present, that fails to show that that microbe actually caused the disease. It has to be present at the end of the injection. Otherwise, something else could have just killed it. By coincidence, you would then be making a mistake. So just as a brief review here of the timeline, I thought it was pertinent to lay it out for you guys like this. So first, the ancient Greeks, remember they proposed miasma, the bad air theory, that bad smells, bad vapors and night air will cause disease. And then it was Frank Castoro. We didn't really talk about him, but he began discussing early versions of the germ theory in his writings. And then Robert Hooke observed cork cells with his microscope. So he found the cells, he started developing microscopes. Van Leeuwenhoek sort of took this microscope idea and ran with it, and he saw many single-celled organisms and made the drawings of them. And then Semmelweis went and remember with the Lyme, he was the guy who worked with the pregnant woman. He showed that hand washing reduced puerperal infections. Here's someone else we didn't talk about, Snow, and funny enough, this was actually a John Snow, not the one that I'm sure you're all familiar with, but this one was probably even more impactful. So he was one of the founders of modern epidemiology. Specifically for people who are interested in water quality, his work is really, really, really amazing. I'd strongly encourage you to go read a bit about him. So he was responsible for tracing the source of a cholera outbreak in Soho in London in 1854. And he actually stopped this cholera outbreak by convincing town officials to remove the handle of a water pump. And then they saw, oh wow, now people are not getting sick anymore. Because in that instance, what was happening is the wastewater was contaminating the drinking water, and then people were extracting that drinking water and just drinking it and getting sick, and no one could understand what was going on. So Snow also his work also inspired the adoption of anesthesia, as well as fundamental changes in water and waste systems of London, specifically keeping water and wastewater separate, drinking water and wastewater separate, which led to similar changes in other cities and a significant improvement in public health around the world. So you can just imagine how many millions of lives his work has saved. And then so after John Snow, we move on to Pasteur with his he finally showed that spontaneous generation of microorganisms was not true, and that you can extend the life of milk and other similar spoilable products by exposing it to heat, so heat can kill microbes. And then Lister here began using carbolic acid to disinfect his instruments during surgery, and then Robert Koch determined the causative agents of bacterial infections and formulates Koch's postulates. So we're moving on to vaccination, currently a big hot topic. I'm sure all of us are very keen to get our hands on the COVID-19 vaccine so that the world can go back to some semblance of normalcy. So I think it's pretty pertinent to talk about this for a couple of slides. So the vaccination was actually started by Edward Jenner, who was a young physician in 1796. So in 1796, he started working with cowpox. How can I? So just what he did was morally questionable at the very least, and it would not pass for how vaccines are developed nowadays at all. But so what he did in 1796, why I'm struggling to articulate this, he carried out a famous experiment on an 8-year-old boy. I think it was one of his farm hands, if memory serves correctly. So what he did is he inserted pus he inserted pus taken from a cowpox pustule and inserted it into an incision on the boy's arm. So he essentially infected the boy with this pustule. Obviously, he weakened it and all of that first, but yeah. So again, not ethical, not the way he should have done it. But luckily, his work then led to this boy becoming immune to cowpox, and then that's what led to the vaccination, which is turned. This is why vaccination comes from the Latin word vacca, which means cow. It's because of Jenner's work with the cowpox and the 8-year-old boy. And the protection provided by vaccination is then called immunity. He was also the pioneer of the smallpox vaccine, which was widely seen as the first vaccine. Again, just I'd like to stress this, people nowadays don't infect children with diseases the way this guy did. So it was morally quite questionable what he did, but luckily it had a positive result because his work did lead to the first vaccine, and the boy, after subsequent testing, the boy did become immune to that disease. So how does vaccination actually work? So vaccinations induce long-term immunity to specific pathogenic infections by stimulating the production of memory cells. A vaccine is a weakened or an attenuated form of the pathogen that contains the antigens, but is incapable of triggering the disease. So you are essentially, even saying a weakened form of the microbe is not correct. So you are injecting someone with something that will elicit the immune response without the possibility of making them sick in most cases. The antigenic determinants in a vaccine may be conjugated to an adjuvant, which functions to boost the immune response. The body responds to an injected vaccine by initiating a primary immune response, which results in memory cells being made. So you can see here, he's being injected and the memory cells are then being made. Keep in mind, important step here, this pathogen is attenuated here, so it's weakened down here is where it loses its ability to cause the disease, and then you this antigenic determinant is combined with the adjuvant, and then that's what you are injected with, and then that is why you get the memory cells being formed. So when you are then subsequently exposed to the actual pathogen, so if you are then, let's use COVID as an example, if you get the COVID vaccine, you already have some immunity to it. So then when you are exposed to the vaccine, to the SARS-CoV-2 virus, you already have a resistance to it. And you have a much stronger immune response to it. So as a consequence of this more potent immune response, the disease symptoms do not develop, so that the individual will then become immune to the pathogen, in this case, COVID-19. So you get the vaccine, your body already starts building up a defense against this microbe. Now, when the microbe arrives at a later date, if you get exposed to it, your body's natural defenses are already so set up that it can't do anything to you, so it becomes harmless. So the length of time a person is immune to infection following a vaccination depends on how long the memory cells survive for. Memory cells may not survive a lifetime, and individuals may subsequently require a booster shot to maintain immunity. And notably, something that also comes up a lot by people who don't necessarily understand how vaccination works, they like to throw the flu vaccine or the flu shot, the annual flu shot. They like to use that as why vaccines don't work. This is, of course, a flawed argument because the flu virus changes a lot genetically. And that's why it is so hard to develop a long-term effective vaccination against it. So every year, the strain of flu that is that season's flu is different from the previous year. And people don't understand that always. So it's very important that when you look at the flu shot, it is an effective vaccine for that year, and that's based on a lot. I'm not going to go into all the detail now, but it's a flawed argument saying that vaccines don't work because why would we need an annual flu shot? It's because the flu virus is not the same. So just going back to some other important things about the history of microbiology. So Koch developed the use of agar to grow bacteria. So agar is a polysaccharide isolated from seaweed. It's a very complex polysaccharide, so most bacteria cannot digest it, and it remains solid over a wide variety of temperatures. So it's a very, very good growth medium. And there was also something else that you guys should definitely be aware of before we start labs next week, is the streak plate method. This is how you isolate a bacteria. So in this case, what you can see here, it's done on agar medium, and we isolate a pure culture of bacteria. So what's done here is you will take your inoculation loop and you will streak some of your sample on the plate, on this section of the plate. Then you will sterilize your loop, and then you will start here without taking more samples and streak this way, making sure to overlap some of your previous streaks there. And then again, you will inoculate your loop, and then not inoculate, you will sterilize your loop and bring it back to your plate, streak across the old ones, and just keep streaking it out, spreading the bacteria. So what that results in, you can see here initially where the person streaked, there's just so much bacteria. But with every subsequent streaking, the abundance of the bacteria decreases until you get the formation of single colonies. And that's how you isolate a bacteria, because you're essentially, you're adding organisms, then you're taking a fraction of those by streaking, and then you're taking a fraction of that fraction, and eventually you just go down to one cell, which is that's an isolated bacterial colony. Okay, so now something else that we should also mention, we will be doing lectures on it later on, so I'm not going to go into very great depth today, but so we can definitely take measures to prevent infections by using vaccines and disinfectant techniques and aseptic techniques, but can we treat it? And we will be discussing Alexander Fleming and his, let's say, accidental discovery and the development of antibiotics. We'll be doing an entire lecture on those, so I just wanted to know that, yeah, obviously we can treat it in certain instances. And then something else that I feel I should just take a couple of minutes talking about here, or maybe a minute or so, is asymptomatic carriers. So I'm sure all of you are aware of Typhoid Mary. She was the first known carrier of she was the first known asymptomatic carrier of typhoid fever. She infected about 50 people before she was quarantined. She served uncooked food, so raw fruit, and transmitted infections because she also just flat out refused to wash her hands before working with the food. And then why this is important, an asymptomatic carrier is a healthy carrier, or it can also just be called a carrier, is a person that asymptomatically carries an infectious pathogen. So they are not showing that they are sick. Very important here. Although unaffected by the disease themselves, carriers can transmit it to others. This is also why in our current pandemic, mask wearing is such an important thing, because people like saying, I don't feel sick, I don't need to wear a mask. But you could potentially still have the disease and just be in the asymptomatic period of the disease, or you could just simply be a carrier, an asymptomatic carrier, and have the disease and never feel sick, but you could still spread it. That's why mask wearing in the current, that's another reason why mask wearing is such an important thing for the current pandemic, and why we should all keep practicing that on campus in your daily lives. So you can, to using the salmonella, or the typhoid fever as an example, scientists calculate that between 1 to 6% of individuals infected with Salmonella typhi, the microbe responsible for causing typhoid fever, become chronic asymptomatic carriers like Mary. So Typhoid Mary was also a strange case because she was quarantined by the government against her will for long periods, and there's a lot of moral questions about that. Like, I mean, they took away her freedom, but she didn't do anything wrong except spread the disease. So obviously there can be a lot of debates about that, but I think the correct decision was made because, you know, she could have potentially infected and killed many more people. Okay, so just some details about the naming and classification of microorganisms, as I just spoke about that. The causative agent of typhoid fever. So this is also known as taxonomy, which is the practice and science of classification. The practice and science of classification of things or concepts, including the principles that underlie such classification. So how do we divide microbes or living organisms into groups based on what characteristics? So Carolus Linnaeus established the system of scientific nomenclature in 1735, and each organism, when we talk to each microbe that we're going to be discussing, will have two names: the genus, you see down there, the genus, and the species. So this is all the way from domain, remember the domains we discussed, and then kingdom, phylum, class, order, family, genus, species. So microbes are always referred to by both genus and species. Here you can see this concept, or how we refer to microbes, is also referred to as binomial nomenclature. So some examples here include Bacillus anthracis, that's the anthrax one. Clostridium botulinum, this is the microbe that actually creates Botox. And interestingly enough, I'm not sure how many of you know this, but Botox is one of the most toxic compounds known to humans. It is ridiculous, way more toxic than something like hydrogen cyanide, for example. And then Escherichia coli or E. coli. So Escherichia here is your genus, coli is your species, and then Streptococcus pyogenes. So you can see you italicize both portions of the name, and then the genus is in uppercase, and then the species is in lowercase. Strain numbers, this is the last one of the last slides, getting to the end here. Strain numbers usually consist of 2 or 3 capital letters designating the lab where they were constructed, and a serial number of the strains in the central laboratory collection. Alternatively, strains can also be named after plasmids or unusual characteristics, etc. So if you look, for example, here at E. coli 0157H7, this is one of the most pathogenic E. coli strains, serotypes, and this is usually the one that causes widespread foodborne outbreaks. It's usually this one, and this 0157 is the plasmid that contains the pathogenic genes. Okay, so today's review questions. Which of the following is not one of Koch's postulates? The microorganism must be re-isolated from the inoculated diseased individual and matched to the original organism. The inoculation of a healthy individual with the cultured microorganism must recapitulate the disease. The microorganism must be present in diseased individuals. The microorganism must be cultured from the diseased individual. And the microorganism must be found in the diseased but not healthy individuals. So I think I'm going to give you some time to think on this one. Notice that the wording I use here is different than in the slides and different than in your textbooks. I will definitely be doing similar things in exams because I like to test your understanding, not just your parroting. I don't care if you can memorize a bunch of facts. I want to know that you are actually understanding what those facts mean. So just, repeating the exact same words in the exact same order without understanding their meaning, to me, it doesn't mean anything. So I reckon that gives you about enough time. I'll give you about 5 more seconds here. So think which one is not one of his postulates. And then the correct answer here is, of course, C. I might have misspoken when I asked this question, because I think I might have said diseased, but it's deceased individuals. So this is not one of his postulates. The microorganism must be present in deceased individuals. I mean, an individual can die from anything. So just because he is the organism is dead doesn't necessarily mean he died from the microbe. The rest of them are his postulates, just in different wording. So who disproved spontaneous generation? Was it Robert Koch? Was it Louis Pasteur? Was it Robert Hooke? Was it John Snow? Or was it Edward Jenner? I'm going to give you less time for this one because this is pretty straightforward. You can always feel free to pause the video if you need some more time. So the correct answer here is, of course, Louis Pasteur. Remember, he disproved the spontaneous generation with his S-shaped flask that he used. He specifically disproved spontaneous generation for microorganisms. So true or false, animalcules were first described by Robert Hooke. I'm going to give you, say, 10 seconds for this. It shouldn't take you that long. Was Robert Hooke the person that described animalcules? And the correct answer here is false. Remember that it was Antonie van Leeuwenhoek that described the animalcules, the guy that had, the merchant that had the like 400 plus microbes. He was the one that described and drew the animalcules. Robert Hooke was the first one to create the microscope, and he looked at the cork cells. And then true or false, question number 4, only people displaying symptoms can spread disease. This is a pretty simple one. I went on quite a bit of a tangent on this, so I'm expecting everyone to get this one right. I'll give you 5 more seconds. And the correct answer, of course, here is false. Remember, you can be an asymptomatic carrier, or you can just be in that portion of the disease where it is not, you're not displaying symptoms yet. So thank you everyone for listening to the lecture again today. We will be looking at some other work next week, and please remember to sign up for the labs in your, sign up for a lab section. That it has to be done by this week, because next week we are starting with labs. And please only show up to the section you signed up for. Don't attend the other sections; it's not needed. And then I will see you guys soon. Thank you so much. Bye.